As mentioned before, the C3 cycle is only one version of the way plants actually fix carbon and turn it into sugars. This happens to be the most broadly used type of dark reactions within plants. But let's talk about the second most popular way to fix carbon, and it is referred to as the C4 rather than the C3 cycle. The big difference is it includes this entire process, the C3 cycle, but it has an additional group of steps, let's say, that precedes this part of the dark reactions, and it makes it more efficient in certain situations. And so when we talk about plants that use C4 photosynthesis, or we call them C4 plants, C4 plants use C3 type dark reactions. And so they have Rubisco and all those other things that we've talked about before. But in addition, they also use another cycle that's called the C4 cycle. And they're named C4 plants because, as it says in your notes, the C4 plants produce a 4-carbon compound known as OAA for short, which is for oxaloacetic acid, which contains 4 carbons. And so that's where this name comes from. But C4 plants have this extra cycle, and they also have some other unique features that separate them from C3 plants. And these are referred to as Cran's anatomy. And let's look at a photo of the cross section of a leaf having Cran's anatomy. And they look a little different than a C3 leaf. And here we are. This is a cross section of a leaf. We've got epidermal cells up here. If you look closely, you can see stoma openings. These are guard cells on either side of these openings. But these are the vascular bundles. In other words, these are the xylem and the phloem in this leaf. And leaves that have Cran's anatomy, i.e. C4 plants, they tend to have certain features. And one is that they have much more numerous veins in their leaves, and so these veins are very close together. They also don't have any spongy mesophyll. If you recall back from looking at our early morphology leaf cross sections, we had what was called spongy mesophyll. These simply have mesophyll cells that are very tightly packed together. Each of these cells contains numerous chloroplasts, as you can see. But the cells that have the most chloroplasts are these larger cells that are clustered very tightly around the vascular bundles, or the veins, basically, and arteries of the leaf. These cells are referred to as bundle sheath cells. In fact, collectively, that layer of these thicker, larger cells are called a bundle sheath. So in this particular case, this is a bundle sheath cell, and they surround the veins. They have more chloroplasts than the typical mesophyll cells. And so basically we've got two different types of chloroplasts going on here. Bundle sheath cells have one type of chloroplast. Mesophyll cells of a C4 plant have a secondary type of chloroplast. So two types of chloroplasts. So that's Cran's anatomy. Bundle sheath cells, no spongy mesophyll, it's got tightly packed mesophyll, and very commonly also are these close together veins. They're spaced very close together. Cran's is not named for some particular person's name. Cran's is German for wreath, and that just refers to this circular arrangement of bundle sheath cells around the vascular tissues within a leaf. And that's where the term Cran's comes from. Now, why have all this extra stuff? Well, basically, C4 photosynthesis is a mechanism for allowing these mesophyll cells to concentrate CO2 into the bundle sheath cells. So let's write that out here. C4 concentrates carbon dioxide into BSC for bundle sheath cell. There's a little hint for you. And so let's look at this a little more closely here with a figure. This figure depicts C4 photosynthesis, and as I said before, there are two different types of cells at work here, each with their own type of chloroplast. Now remember, we're talking about cells with chloroplasts inside of them. So these little green circles represent a chloroplast. This is certainly not to scale. Chloroplasts are much smaller than the overall cell than they're showing here in this drawing, but I think you get the idea. So we've got mesophyll cells, which are very tightly packed together, and we've got these bundle sheath cells. And immediately inside of the bundle sheath cells, although they're not drawn in here, would be the xylem and the phloem, in other words, the vascular cells, or the veins, so to speak. And so how does this work? Well, along here would be another bundle sheath cell, and over on this side would be an additional bundle sheath cell, making up the crans, or the ring around the vascular tissue. So carbon dioxide comes in through the stomata, and the mesophyll cells absorb it. When they do, instead of Rubisco, they use an enzyme called PEP carboxylase, which carboxylates a molecule called PEP for short. It's something like phosphoenol pyruvate. Just know this PEP carboxylase is not Rubisco. It basically does the same thing as Rubisco by fixing carbon, or carboxylating PEP instead of RUBP in the C3 cycle. But at any rate, the first thing it gets produced is OAA, which again is the 4-carbon molecule, OAA. It also uses a little bit of NADPH initially to turn that into an organic acid called malic acid. Now, malic acid is very soluble in water, and it's easily transported between cells, and so it gets transported from the mesophyll cells into the bundle sheath cell. Now, once inside of the bundle sheath cell, this NADP that was added gets taken out of it through an enzymatic reaction to release CO2. Then what's left over is called pyruvic acid, another organic acid, which gets transported back to the C4 cells, i.e. the mesophyll cells, where it is used to regenerate PEP and keep this C4 cycle going. Now, this step requires an additional ATP, and this is important. I'm going to come back to this in a few minutes. The rest of the story is basically just like C3 photosynthesis that we've talked about. The CO2 is used by Rubisco to carboxylate RUBP and turn a series of other reactions to end up making sugar. And so you're looking at this, and if you're thinking about it at all, you're probably saying to yourself, well, what's the point in all this other mess up here? Why would a plant have all of this extra stuff going on? And in addition to that, it actually costs more energy, if we think about it, to make the same sugar molecule. A C4 plant, all other things being equal, will require 2 extra ATPs, which come from the light reaction, just to run the C4 cycle. So C3 alone is running on the same amount of ATP as we discussed earlier. Back to my question I just posed a minute ago, why would a C4 plant go through this trouble? To answer our questions in terms of C3 versus C4 photosynthesis and which is better, we'll talk about that in the next module. We'll explicitly describe why in some cases plants that have these extra steps end up being much better at making sugar than the plain old regular C3 plants.